seen from this couple of lectures. And these I haven't shown you also for like different sessions where here you have mitosis and here you have meiosis. So make sure you know these couple of slides, really well, okay? What are they? Okay, so here you have meiosis, of course, in germ cells, mitosis, of course, in somatic cells, cells in the body, where you have cytokinesis that occurs once in mitosis, but twice in meiosis. And in mitosis, the daughter cells will have the same chromosome number as the parent cell, so they're genetically identical. It is equational. Whereas meiosis will have the chromosome number is going to be half, so they're going to be reduction. So you go from diploid to haploid. Okay. Mitosis, 2 daughter cells. Meiosis, 4 daughter cells. Mitosis, genetically identical. Meiosis, they're going to be different. Okay. There's going to be genetic variation. They're going to be different among the cells and different from the parent cells. So also the mechanism of action, make sure you know that between mitosis and meiosis, that in meiosis 1, here in prophase, you have carcinoma, okay, homologous chromosomes that are going to grow up next to each other, and they're going to exchange this genetic information, carcinoma, okay, charismatic. And also in the metaphase 1, those homologous chromosomes are going to pair next to each other, double file, and then in anaphase 1, those homologous chromosomes that will separate. And meiosis 2, similar, not identical, but similar to mitosis, in the sense that the sister chromatids don't separate. They are different in the sense that the sister chromatids, they are not going to be identical as it is in mitosis. Okay? So make sure you know those key components. Why do you use mitosis in the organisms, in the body, right? That will be for growth and development, tissue renewal and repair. In single cell organisms, asexual reproduction. And here, there will be meiosis in organisms in humans, for example, to generate those sperm cells, those egg cells, to produce the gametes, genetic variation. We mentioned this as well, a couple of sections, that Mendel's hereditary factors, now we know them as genes, and we know that they're located in chromosomes, and they have the specific locations, the loci in those chromosomes. And these genes will have like hundreds or thousands of these genes. For example, here you have this chromosome that is unduplicated, and then you have here this gene 1, 2, 3, 4, 5, 6 genes right there. Whereas here it's been duplicated, so now you have the same thing, 6 genes, but now here you have 2 alleles, 2 alleles, 2 alleles, so 6 genes, 12 alleles. So make sure you keep that in track. Here, just so you can see, in a chromosome can contain hundreds or thousands of genes, and each one of them they have a specific location. So here, for example, you have, let's focus here on 2 genes. Each chromosome carries an allele, so that's right here, right there. And the alleles on a pair of homologous chromosomes can be the same or different. Right? It can be both homozygous or it can be heterozygous, okay? These are the linked genes in case they're like really close together. And these genes, compared to that one, they are really far apart, so they're going to be assorted independently, okay? Or this one as well, independently. If they are really close together, then they are linked genes, so they will behave as one. So that's what we will see, that ratio of dependence of. For other genes who are far apart on the same chromosome or from other chromosomes, then that will be independent as well. All right, so we saw this as well from last time, the parental generation, where you have here the yellow round seeds dominant for both genes, dominant or for alleles, the yellow dominant color and round seed, the shade, both dominant, and here double recessive as well. The green recessive and the wrinkle shade recessive. Okay, so these are true breeding. You cannot give any other allele but that to give that particular trait. Yellow and round and green and wrinkle. Okay. Then you have here the gamete production for this. This one cannot give any other than this, the capital R and the capital Y. Same thing here. This one cannot give other than the recessive alleles to produce this green and wrinkle. That is parental generation. Then you have the F1 generation. So once you have here the parental generation being homozygous, dominant, recessive, both of them. Then you have the mixture of that. You have the hybrid generation, the F1 generation. So that will be heterozygous. Okay, but because it's complete dominant, that's what you will see. Okay, both dominant, color and seed shape, yellow and round. That's what you see. But it carries the allele that are recessive for this trait. Okay, that's what you see right here. Now from here, make sure you are able to know how to determine the gamete production. Okay, so you have here these two genes, one gene, another gene, that's 1, 2, 4 alleles right here, these and these. So 2 alleles, 2 alleles. Make sure you're able to know how to determine the gamete production. Okay, so here you have these. Let's say this is your F1 generation, right? To produce the F2 generation, so you have the gamete production, and these can give these and these, so that would be big Y, big R, and these, big Y, little r. For the same time, this little y can go with this big R, and it can go with this little r. Okay, so you have those 4 gamete production. 1, 2, 3, 4. Okay, so make sure you're able to determine those gamete production based on that genotype. Right, so and that's what you have right here. And this happens during these metaphase 1, anaphase 1, and then metaphase